I would like to present the plans for SP2 dealing with trophic interaction, tree diversity and soil erosion effects on soil-plant stoichiometry. This project will be led by Steffen Seitz, Thomas Scholten and myself, and we will closely collaborate with our Chinese partners Naili Jiang and Yu Liang. Previous work highlights the role of organisms for soil erosion. As you can see on the left-hand side, with increasing tree species richness, soil erosion decreased, and this effect became more pronounced with ongoing experimental duration. Similarly, on the right-hand side, it is indicated that the presence of soil fauna increased soil erosion. Therefore, there is an effect of organisms on the distribution of masses, and the question now is whether this also applies for the redistribution and partitioning of nutrients. Topography acts as a strong driver of soil erosion, and tree diversity might modify topographic effects, and both might jointly influence the redistribution of nutrients along slopes and thus separate soil fertility. The resulting modified soil fertility as a resource for organisms likely feeds back on other trophic levels. And similarly, as has been shown for a forest ecosystem, the environment to which also soil fertility as a resource for organisms belongs was shown to have bottom-up effects. Previous sub-projects have already highlighted the positive effect of tree diversity on the productivity of primary producers, that is, of trees. If trees grow better, they also need more nutrients, and the increased nutrient demand can either be met by more efficient nutrient recycling, meaning that organisms preserve nutrients while growth increases, which will shift the stoichiometry towards the growth-related element, which is carbon. Alternatively, the increased nutrient demand can be met by increased nutrient exploitation, that is, increased nutrient uptake, and accordingly carbon to nutrient ratios will be shifted towards nutrient ratios. Overall, this will mean an influence on organismic stoichiometry. Increased nutrient exploitation has been proven for soil microorganisms in a grassland biodiversity experiment. However, it remains unclear whether this is transferable to more long-lived and complex forest ecosystems. Similarly, it is not known whether primary producers and higher trophic levels share the same processes underlying tree diversity effects or whether they differ in their response to tree diversity. And this might not only affect one trophic level but propagate into higher trophic levels, which will not be presented in this sub-project but will be jointly studied together with sub-project 3.
Adding to this state of the art, there was more preliminary work already conducted by the applicants together with other sub-projects within BEF China, and leading to more than 40 publications in total so far. Moreover, a soil baseline sampling was started in 2010 and data used by many partners in manifold topics. This brings us to our objectives, which we organized in 5 work packages. In a first step, we plan to provide a new soil data baseline investigation and enhance the existing timeline as a courtesy to all sub-projects. In this first work package, we will conduct an early project sampling campaign together with SP3 on all core plots with follow-up soil analysis in the lab. Subsequently, and to connect to the aforementioned state of research, we then propose 4 hypotheses which are presented as work packages 2 to 5. Our first hypothesis takes into account that mechanisms of soil erosion can redistribute nutrients along slopes and differentiate nutrient patterns within soils, as you can see here. We assume that the stoichiometry of the eroded material changes during transport and can translate into higher trophic levels via soil microorganisms and tree roots. Thus, we want to investigate the differentiation of nutrient patterns by erosion using experimental field techniques with runoff plots of different sizes, stem flow gutters, and multi-elemental stoichiometry. For the second hypothesis, we assume that eroded slope positions show lower nutrient availability and foster nutrient recycling, while deposition areas are dominated by nutrient over-yielding. In contrast, diverse tree mixtures might balance the trade-off between nutrient uptake and nutrient recycling by mitigating soil erosion and offsetting topographic differences. Thus, we will use our broad soil dataset to model nutrient availability and recycling on a hill slope scale, including core plots on all slope positions with different diversity levels and using state-of-the-art techniques for machine learning and end-member modeling. This study will be in close cooperation with the Machine Learning Cluster of Excellence at Tübingen University. Thirdly, we expect that, similar to evidence from grassland biodiversity experiments, soil microorganisms increase nutrient exploitation, which will result in nutrient over-yielding. Accordingly, C nutrient ratios will be shifted towards nutrients and thus decrease with increasing tree diversity, as it is shown here. Therefore, we will use multi-elemental stoichiometry of soil microorganisms and directly link stoichiometry to underlying tree diversity effects. Nutrient over-yielding of soil microorganisms will be studied based on microbial elemental stocks as a product of element concentration and microbial biomass. As recycling within soil microorganisms is challenging to study, and particularly for the large number of plots here, we will approach recycling using microbially cycled nitrogen and phosphorus that is left over by soil microorganisms. To this end, we will use an oxygen isotope approach. Finally, we postulate that trees will rely more on nutrient recycling to meet the increased nutrient demand induced by tree diversity. Therefore, the response of different trophic levels to tree diversity in terms of nutrient cycling will differ. Thus, we will assess multi-elemental foliar stoichiometry of trees. Foliar nutrient stocks will build the basis to calculate nutrient over-yielding. The ratio between nutrient concentrations in senescent as compared to fresh leaves will be used as a measure of resorption. Regarding networking and integration of our project within the research unit, SP2 will link to the modeling of trophic interaction networks, SET1, as well as to associated multiple functions, SET2. Further, we will provide multi-elemental stoichiometry data for soils, microorganisms and trees, and biomass and element content data towards higher trophic levels, for example herbivores and predators in SP1, 3, and 6. Last but not least, we provide baseline soil data of very dynamic soil properties like soil organic carbon, pH, and nutrients to all subprojects of multi-troph. Having said this, our project will contribute to the construction of large multi-trophic food webs and here to a better understanding of effects of nutrient recycling, exploitation, and redistribution along slopes, for example by erosion, on associated multifunctionality, and on overall tree performance and biodiversity. To do so, we receive process data from SET1 and SET2 that reflects findings from all subprojects and, more specifically, organismic data from deadwood decomposers from SP1, predators from SP3, and ants from SP6, who nest in ground and can see or perceive soil quality. Finally, Naili Zhang and Yu Yang are the principal investigators of our Chinese partner projects. Both have secured funding from Chinese Academy of Sciences or the National Science Foundation of China And their research aims on effects of tree diversity on pathogens, led by Yu Liang, and the role of the microbiome in regulating plant soil feedbacks, led by Naili Chang. Having said this, we can now go on to the next presentation of SP3, given by Jana Petermann.